So there I wrap up the lecture that we essentially did on the whiteboard, so the prediction of sub-cellular localization. And on Thursday, in fact this has to change. Tobias is not going to do the lecture on Thursday. Is Tanya going to do that? Because Tobias is sick. And next week we may still go on sub-cellular localization. So the de novo prediction of sub-cellular localization. Let's begin with the words again. Most of you have seen these words. What's the difference? The meaning. Somebody who hasn't seen it. I assume you have seen it, Sabrina, don't worry. So you're off the hook. Anybody who hasn't seen it? What do you associate? Let's begin here. Ab initio. What does it mean for you? So, anybody? You. So the newcomers. So okay, let's just jump into it. Ab initio is really the idea that you have first principles. You can write down an equation. From these first equations, you can make a prediction. De novo is the idea that you make a prediction without using existing information, or not directly at least. That becomes more clear if I compare it to by homology. Homology-based inference is really such that I know and have experimental evidence for a particular protein having a certain function in one organism, and I use a similarity to my protein of unknown function in order to infer that the two have similar function. Typically I can do that only because they are evolutionarily related. Typically, at whatever level of sequence identity we're talking about, 60%, if there would be random sequences not evolved under constraints of life, maintaining function, then most of the cases would not reveal functional similarities. They do that only because of the statistical background signal being so strong that they do that only because, in fact, there's a constraint built into the sequences that we see today. That's why why I insist on this term homology, to make it clear to you there's really necessary an evolutionary relation, and that in fact means I know experimentally that one similar to that one, I can infer the function. That is the type of meta-student story that you were presented with. De novo is the type of machine learning device where you extract some general principle, some general features, but you cannot quite say these general features tell you that, or you cannot say this is because these two proteins are similar and I have an experimental annotation. There's something in many proteins that for which you have an experimental annotation, and that feature helps you to make a de novo prediction. Now the problem is clear. So we have a prokaryotic cell, a eukaryotic cell. We want to distinguish different types of sub-cellular localization, and we want to do that because sub-cellular localization tells us something about the function and is something that we can easily measure. There are different resolutions, and we begin with a relatively simple one. There's a pathway of trafficking in the cell. There's a direct exchange between the cytosol and nucleus, and then there are these green compartments here and the blue ones. So the blue ones, essentially all of this is the secretory pathway. You go to the endoplasmatic reticulum. Whether you go on or not depends on whether you have a retention motif. If you have a retention motif, that's the way we believe it. Most retention motifs are not known. But the current assumption of biology is that the ones stay in the ER that do have a retention motif. Again, for most of the things in the ER, we do not know that motif. But that's the current assumption. The same story happens, it goes on to the Golgi, for the ones that don't have the signal. They go into the Golgi. Again, we have a retention motif, a lysosomal retention motif, and ultimately towards secretion. Some proteins stay in the membrane, some proteins really get out. This is gated here in red, and that, by the way, is a picture from the Alberts, the cell. This is transmembrane transport, and that is sort of vesicular transport, secretory pathway. Now, the way we can predict sub-cellular localization is by similarity. We talked about that, and I'll get back to it. By text similarity, by motifs, and de novo. Homology-based inference. And that is what we had in the lectures before. You measure the similarity between two proteins. The work goes back to Rajesh. And that, just to remind you, you have to find a sequence-unique subset. By that, you simply take the Swiss prot annotation. Now, there are different ways in which we can establish sequence similarity. Percentage sequence identity. That was the alignment length versus percentage sequence identity. Last time I showed you the graph, it had different colors. So last time the red were the mismatches and the blue were the matches. Here now the green are the mismatches here, and the red are the matches. That's percentage sequence identity. Then there is an h-val, here's this p-value. Some of you have seen that in the last lecture. Essentially there is this blue curve here that is such that most proteins above that blue curve have naturally evolved to have a similar structure. And now you can simply take this curve and compile it every single position, and you see that in this curve it measures how long the alignment is, so very short alignments, 11 residues. You cannot tell anything about structure. 100% identical doesn't help you, because two 11 mers can, in different structural contexts, adopt different functions, different structures. And then the longer the alignment gets, the less sequence identity you need. It sort of saturates around 20% for very long alignments. Now you can simply rank all my points according to the distance from this curve. So this here, this blue is +5, +10, and so forth, and that is then would be a minus value, right? Below the curve. And so then we can essentially measure in terms of HSP distance here, that is the value that I talked about, 0 for long alignments, 20% sequence identity, how sub-cellular localization is conserved for different classes. There's something else we can measure sequence similarity by, namely the most familiar one to most of you, I guess. Percentage sequence identity is something that everybody uses because it's simple. H-val is very peculiar or particular. It's very successful, but very few groups use it. RMSD? Yeah, not a sequence value, right? RMSD doesn't help you for sequences. Essentially, if you would make your statement, if you would argue, then you could compile an RMSD-like value, and that would be somehow similar to sequence identity. But this is a push of the interpretation.
Alignment score?
Yeah, an alignment score or something similar to an alignment score, which is a significance value. So the E-value from BLAST is not in fact an alignment score, it's a significance score, and the significance score always is defined with respect to some background. Blue, the findings that you want to evaluate, and essentially you want to measure how much that score that you see there differs from the background. So this would then be the raw alignment score that you talk about. And so the degree to which it is unlikely that it has arisen a chance has to do with how far that is from the mean and how much this curve spreads, right? These two values, so essentially the shape, the two main values of the Gaussian the Gaussian-like distribution enter the answer of how significant a particular alignment score is. So this raw score is very informative, but against the background is getting more informative. That's the PSI-BLAST E-value that you see, and then you in fact get different. It's not about the details here. I just want to show you that some of the details that I didn't show last time I showed this concept. So now we have the HSP value, the E value, the percentage sequence identity. Here we go with accuracy or a percentage value. In fact, there are always coverage and accuracy curves here. So for every for a particular threshold, and percentage sequence identity, you see where you get, I believe, a note here, 95%. The errors are 95%, and the degree to which of these three would be best. How would you decide the answer to that? So one criterion is easy to compile, and that certainly is here. But that's not what I mean. Which of those is most successful? How would you decide? You have to. One way is you put yourself on one of these curves here, such as this arrow, and you ask the corresponding value here in terms of coverage. So if I use PID, percentage sequence identity, at 95% accuracy, what I get is about 50% coverage. Here I get to a value that is significantly higher. E-value is much better percentage sequence identity is what we learned from that. That's pairwise blast up there. And here, again, I assume that this value should be higher even. I'm not entirely sure. Yes, I see this is even higher. The story changes slightly when you, instead of looking at pairwise alignments, look at a psi-blast alignment. When you look at the power, when you benefit from the power of the profile. These, in fact, are still not ROC curve, mind you. ROC curve would really look at cumulative accuracy versus cumulative coverage. And in fact, so that's another way in which you can say or can distinguish between different thresholds. So you could argue that the psi-blast e-value here is better because there's a region where it is clearly higher than the others. You could argue that the h-val here is best because it has the highest area under the curve. But again, so there are different ways of seeing that. This is the multitude of data. What you clearly, although you don't quite clearly see it on this slide, due to the quality and to the amount of data that I show on this plot, but I believe you can all appreciate that black is always higher than gray. Gray is the pairwise blast, black is the psi-blast, and for whatever you look at, that is higher, so benefiting from a profile from psi-blast is always good advice. Back to the story, that is structure, that is sub-cellular localization, and overall in perspective, essentially, let's call it 4 different features: green, similarity in structure, by HSSP or H value, and by the blast E value. Blue, the degree to which you conserve subcellular localization. In this particular case, in 4 classes. Enzymatic activity, the orange here is the point where you conserve the first EC number. Red is all 4 EC numbers, and you see red deviates much earlier than orange. Now, what we observe in all of these curves, we see that best here, somehow see it here, but somehow clearly better, is that there is an extremely strong drop. Why is that? Physicists would call such a drop zone a phase transition. So you go from a regime, let's not look at the differences, let's look at the similarities. The similarities between all 4 curves are that there's an area here, somewhere, it depends a little bit the way you define it, where mostly you have high signal, high accuracy, so mostly two proteins, if they have that level of sequence similarity, are similar by the feature, by enzymatic activity, by subcellular localization, or by structure. And then we go within a few moves of my sequence similarity here, we go into an area where essentially we have random signals. So subcellular localization, remember, is 4 states, so this is close to the random for 4 states. That's why it ends up there. But why is that? Why is there such a phase transition type of behavior? Is that trivial? Do you believe this is something that has to do with biology? Or do you believe it's something that has to do with sequences?
I believe this has to do something with sequences, not with biology.
Why?
Because the signal is lost.
Why is that? But so let's begin. Let me help you with the argument. So I would argue, and I can punch a hole in my own argument, but in support of your argument, I would argue that we actually look at fairly different features here: subcellular localization and structure. And the assumption that they sort of collapse at a similar point is not intuitive at all. And even looking at enzymatic activity, you somehow see a collapse in the same region. And the biology underneath the conservation of structure and underneath the conservation of enzymatic activity is very different. Hence, If I see the similarity, most likely this must be a sequence signature or sequence feature, because essentially I cannot explain it by biophysics. That would be the argument in favor of your belief. And in fact, if we simply ask, what's the probability of randomly superposing two strings of letters with a certain frequency, then we do see, it depends a little bit on your assumption of the way you do alignments, the way you compile the score, the way you have gap open and gap elongation penalties, where exactly the transition will happen. But you can absolutely show that without assuming anything about biology, just the fact that you have 20 letters gives you a phase transition. And ultimately this is a statistical signal of the sequences. Okay? So somewhere here, wherever that is, somewhere in this region, what we are seeing is the end of the possibility to capture the similarity, because you just go against the ocean of possibilities. It's just, quote unquote, statistics of sequences. And that is an important thing to remember, at least on your level. Now, at this point, so I showed you more this time about homology inference of sub-cellular localization than last time. What else, if you have enough material for a publication, what else would you do? What else would you ask? What else would you put into the paper in order to convince readers? So you have all these curves here, you have exactly an evaluation how good it works at a given percentage sequence identity, so you can have a database of things that are similar. You can define your threshold according to that. You can give back to the user exactly your score for probability of being right. And you have a simple method that uses PsyBlast to infer by homology sub-cellular localization, right? Now, what else would you do to sort of spice up your manuscript? Could you do? What additional test could you do? You have to, yeah?
Try on the real data and to see how many annotations can we get with our method.
Exactly. I'm not entirely sure, so your explanation leaves room for understanding and misunderstanding and understanding in a different direction, but I assume what you meant is the following. What you meant is, in fact, you take entirely sequenced organisms as one: yeast, fly, C. elegans, Arabidopsis, and human. This is an old slide. So today the story is very different. And you essentially ask, by this method here, the blue, what fraction? So green is where the amount of experimental annotations I had 12 years ago. And blue is essentially what I add by this homology signal. I believe this threshold was taken such that we had 75% accuracy. And that, in fact, is an important statement. So now the question is that impressive? Well, it depends on where you look. So you do much more for human than for yeast. Again, today the numbers would be different. Why would that be the case, you believe? Why is the green, why is the blue relatively smaller than the green compared between yeast and human? It's very obvious. The answer is very simple.
Because in yeast you have more experimental results.
Exactly. It's much easier to do high throughput experiments in yeast, or all kinds of experiments. Many experiments are done in yeast. First done in yeast. And then actually in many of these cases they are done in yeast because people believe that the protein behaves similar in yeast. That's why you can use yeast. Yeast biology is simpler than human biology, and even better would be a E. coli or something for sub-cellular localization. That doesn't work because we don't have the same compartments, but that explains most likely this. Okay, let's get back into a different story, the text analysis story. The background here is the following. What we want to do, again, we want to, in this particular case, try to distinguish 4 different sub-cellular localizations. At the time we did this, 12 years ago, we had about 14,000 annotated proteins overall. Most of those in these 5 compartments, that's why I took them out. And what you in a typical SwissProt record have is a place that says sub-cellular localization, nuclear in this particular case. And then you have a functional. Does anybody see how this function immediately implies nuclear?
Because it's a transcription.
It's a transcription factor and they are only in the nucleus. So this is for somebody who has studied enough biology to make that point very clear, that this one immediately gives that prediction. So, yeah?
Question. But for the transcription factor, it's sub-cellular location content, I think. In nucleus or outside?
Yes. So now we get into an important issue here, caveat in all of the discussion that we have. I look at the conservation of sub-cellular localization, I look at the prediction of sub-cellular localization, yet we know that many proteins are shuttled. Many proteins move between compartments. Now my assumption is, and I have a lot of data to back up that assumption, that justifies it. But it's a little bit of a longer story. But the assumption here is that most proteins are native to one particular subcellular localization. Transcription factor is the archetype of this example. So transcription factors are in principle native to the nucleus. That's where they spend most of the time, where they do most of their action. Okay? And in fact, when you look at, we get back to that later, the surface signal, that is the native subcellular localization signal. Okay? Now, assume Swissprot would not have this annotation. In this particular case, again, as we immediately saw from Sabrina, with some expertise, you can just from parsing this infer that. How can you automate that? And that essentially is, again, work from Rajesh Nair. And so sometimes it is clear. So DNA binding is nuclear, chromatin regulator, blood coagulation, those are kind of clear. But clear means for somebody who knows something about blood coagulation. If you're a computer scientist, you may not know that this is secreted. That there's an outside and inside is obvious for you. So the first goal is to in fact find trivial connections by machines. The second one is to find non-obvious correlations. Okay? How would you do that? So again, oops, the wrong button. You have your 15,000 proteins that have annotations. They all have keywords. You can extract them. So here again is the slide that I showed before. But how would you use that for a method? How would you extract those keywords and build a method that can use those keywords to infer or predict subcellular localization?
Find common keywords in the different subcellular compartments. And cluster them, perhaps. So
you would essentially do an exhaustive search.
Yeah.
Now, your logic implies that there are some keywords, such as DNA binding, that always relate to one particular subcellular localization, and I essentially, by clustering, find those. So you would do well by this. What about this one? Would you discover non-trivial ones? So let's look at individual keywords that are not alone. At examples where you need 3 or 4 different keywords come together in order to, that you could not do, right?
Depends on the cluster algorithm. Because there are some which can detect correlations between the features.
Yeah, you could imagine a machine learning algorithm that could do that. That already is, however, not that trivial. Here's Rajesh, as a physicist, came to a much simpler solution. He simply cut out the obvious and the blah 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 A, which you see because it's present in too many subcellular localizations, and then whatever is left, he built a vector. Okay, the vector is simply, that's the protein. Those are the keywords here, 4, 5, 6, and so forth. And you simply say apoptosis, the keyword is there, calcium binding, the keyword is there, exonuclease, the keyword is there, schizo 1. This is a vector. At the moment I have that, you immediately see, so I take the vectors out of my experimental database, and I build a keyword vector for my query, And then I simply go to my database and ask this keyword vector, which one is that most similar to? Okay? It's a very, very simple idea. You have to work a little bit on what you call similar, but similarity of two vectors is something that is relatively well defined. So some Shannon entropy is an easy way to do that. And you can essentially map by this system any keyword that you have to a prediction. And those are, in fact, kind of examples that in a combination, so that something is a coil does not tell you cytoplasmic, but in various combinations in which they occurred, these were informative keywords. And that shows you that exactly this part of the method worked. It worked overall fairly well when there were enough keywords. And some of these correlations that were discovered were really non-trivial. Okay, then again you apply that to an entire genome, and that was the one that I suppressed before. So the blue is homology, this is for the entire switchboard in this particular case here, and that is the keyword base. So in fact this method is fairly accurate and has high coverage. So it turned out, which we did not realize before, that there were many records in switchboard at the time that in fact did not have the annotation explicitly spelled out because the annotator had simply forgotten about it, because it had been so obvious or had been so clear, and that was a simple way to filter through that. It was a very successful method, and again in the case. So now we get into the issue of motifs. The first issue of motifs, so short stretches, again, they are linear and they can be conformational. We want to find these motifs in order to predict sub-cellular localization, and we remember that this is the trafficking of the cell. Now, we talked about nuclear localization signals. Let's talk about some other motif, signaling peptides. Anybody knows anything about signaling peptides? There's a database? Oh, what did you say?
A little bit. I know a little bit of signal peptides.
Hi.
For example, they are hydrophobic, and it's hard to distinguish from membrane.
Okay, so let me cut to the chase so that you don't give the entire lecture. Essentially, you said everything that I will say, nothing. I'm not going to say anything more. I'm just going to use more words. But essentially, you have the three main points. Secretary pathway. The person who started off this is. Oh, I have to write my email. Is Gunnar van Heijnen. This is from 2010. Very visible. Got a medal. At this point here you see him announcing the Chemistry Nobel Prize in 2008, and he's, I believe, as of this year back on the Committee for Chemistry as the secretary. Gunnar really introduced the concept of how signal peptides regulate transport as one of the many things. Another person who came in the game here is Sam Brunach. Another person who is very highly quoted, star in the field, very known for introduction of machine learning. And essentially these two here, Gunnar knew the biology in the background, Søren is the machine learning expert, and Henrik somehow brought these two together. And that created Signal P, the first version. Now Signal P began on the assumption that signal peptides, as you said, are N-terminal, typically something like 15 to 30 residues. Often they are cleaved during the translocation across the membrane, so they are on the protein until it gets to the extracellular domain, and just then it's cleaved. And they exist in all three kingdoms, so archaea, bacteria, and eukaryotes. And they are relatively simple in terms of the architecture. What it means is there's an N-terminal region that is a recognizable signal is what this means. There's a hydrophobic region, there's a cleavage region, pre-cleavage region, and there's a cleavage site. So all of these things here, the C region here is polar and charged. You essentially deduce all of these informations by aligning, with respect to the cleavage site, different gram-positive bacteria, gram-negative bacteria, and eukaryotes, the alphabet of the letters. And then you see certain prevalences, you can extract that, and you can use that in a machine learning device to simply train signal peptides. You can use neural networks for that. You can use hidden Markov models later. You can combine with other methods. Target P, I'll get back to that in a minute. And you can add additional signals and additional detail. There are many, yeah?
Question, how are these signal peptides attached to the proteins? Are they part of the protein chain?
So when you look at the gene sequence, starting with the methionine in many cases, that's right where the signal peptide starts.
And then there can be a number of different proteins at the other end of the signal peptide.
Yes, absolutely. So now we have to be a little bit careful. I talked about this architecture here. And it's that architecture that is conserved. It's not like in the NLS where it's KL, KD or something like that. KDL, the maintenance for ER or something like that. Here, it is the theme that is conserved. They're all N-terminal. They have a very hydrophobic region, which in fact often, we'll get back to that in a moment, has a reason. There's the C region, there's a cleavage site. Now, typically it says some of these cleavage sites are 70 residues up. And the signal does differ between within different kingdoms. The architecture is somehow conserved. The details are not, and in fact this is not at all the type of signal that is a motif. That is not the prosite motif. It evolved as a prosite motif, but then it got into machine learning, and that was the major step ahead, that realized that this commonality between these, let's call it the architecture here, is good enough for machine learning. Now, you may by now have some idea about hidden Markov model and neural networks. We'll have a flash on these two, on at least the neural network part. Hidden Markov models lend themselves to really putting grammar into the story. So the grammar that I just described, there's an N-terminal region, there's a hydrophobic region, there's a cleavage site, there's a region before the cleavage site. That sort of thing fits perfectly to hidden Markov models. That's why they got into that. And again, as I said, there are many, many other methods available, but so far signal P wins the day. You also made this point of the problems with membrane predictions. In fact, this is a slightly outdated result. Jonas has much later results, and I should actually present it, but essentially this is true here, where we see some methods that have a certain level of accuracy here for predicting transmembrane methods, distinguishing between transmembrane proteins and other proteins. So the error rate here is 1. Some methods are fairly poor, but some methods are very good. Now, however, when you look at the mistake in identifying signal peptides, you see that in fact most simple methods have a high error rate in the 20s and higher. And the reason for that is simply the hydrophobic region that has the length of a membrane helix and the composition of a membrane helix that just looks to all of these methods like a membrane helix. In fact, in many cases, the signal peptide is not cleaved, but it's inserted as a membrane helix. That makes it really complicated to distinguish. A simple hack to this is whenever you predict only one membrane helix and the C-terminal, N-terminal within 50 residues, you call it signal peptide and not membrane helix. That simple hack handles, in fact, most of these problems. A more advanced way, and there's no method up here, but in the data from Jonas, more advanced methods shown here, Phoebus and Spoctopus, are two examples of that. They in fact train the neural network and the signal peptide prediction together as one machine, and then you can in fact do much better than this hack. Okay, are there other signal peptides? So far, mind you, and I put it into parentheses because they will in fact be called something else, but so far I only talked about secretion. Okay? Do you know of anything that is like that in other compartments? So the answer is, that's this one here. There's a similar thing for chloroplasts. Then they unfortunately, welcome to biology, are no longer called signal peptides. Everything is the same. I mean, again, the detailed composition, just like in your question. This is not like a prosite motif. The general grammar for the chloroplast is the same, except for they're now called transit peptides. Same thing. Mitochondria, targeting peptides. Again, same thing. Essentially the grammar is the same. The methods here, chloro-P, mito-P, comes from the same people. Gunnar van Heijnen involved in it, Sir Brunach involved in it. Target P is a method that sort of does this, and that in one method. But then they realized that actually the development of that ultimately for the task of predicting secreted proteins, this is in fact better, because simply this finds a compromise between all three of them. And a compromise is always worse than the best specialist. So that's why essentially signal P and target P both keep on having an own life, and the more better up to date is typically signal P. Here are ER Golgi retention signals, and I already told you they don't work very well. Nuclear localization signal I told you work extremely well, and that again is the story I told you before. But here's another piece. Now we have many nuclear proteins, and in the cell here are some nuclear proteins that have not been annotated and that were related to the nuclear localization signal that we described. And when we looked at the nuclear localization signal, we saw that this was the nuclear localization signal. What is your idea when you see this image? So this is our NLS. This is the signal that in fact we described before regulates the import into the nucleus. So importing, transporting one of these proteins binds to that signal, recognizes that as something that as a protein that has to be imported into the nucleus, shuttles it through the nuclear pore into the nucleus. This is a structure simply highlighting what we know is the nuclear localization signal, that of the type of thing. Remember I told you, you put that NLS onto gold, gold is imported into the nucleus. This is not gold, this is a protein. What do you see? The obvious, just say the obvious. Without using Google. Nobody wants to say the obvious? Okay. Yeah, that's true. Yeah. So this is DNA, right? So to me, this immediately sort of gives the idea that the NLS is also the binding motif. Because this looks just like the binding site for the DNA. Now you have what evolves is an amazing model. You see a nuclear localization signal in the protein, you bind to it, you import this protein to the nucleus, you go off, the importing goes off. The same signal binds DNA. And that's a perfect cycle because many proteins in the nucleus bind DNA. It goes off the DNA, and exporting, that part of the story I don't tell you, binds to the same site again and gets it out of the nucleus. New cycle. Perfect cycle, and for some proteins that works. What we discovered too in this context was that only some of these nuclear localization signal bind DNA. In fact, only about a third. And these could be used to predict new DNA binding sites, which again then we do for Swissprot and we do for other genomes. And in fact those predictions were very accurate. So now let's get back to the de novo prediction, so the overall signal de novo. Now we don't use, so far, signal, overall sequence similarity, all of this is mapping it to proteins for which I know the sub-cellular localization. NLS is an extreme way that is slightly different. Signal peptide in some sense is different. Signal peptide is in fact de novo already, so I mislabeled that. But de novo, let's just extract the principles by a prediction system, by using machine learning, and let's begin with a simple one where we have three different classes. And the first publication, in fact this is not machine learning, this is much simpler. Uses the amino acid composition in the on whiteboard lecture. Sabrina, it was your idea. Using amino acid composition comes back to this paper, and I'm sorry that I don't have. So Kenta Nakai and Kanisha, Minoru Kanisha. I should clearly put up the slides of these two gentlemen, and I'm not sure why they have been suppressed here. Does anybody know anything about Kanisha? Kenta Nakai went on and did many other things. This is just one of his important publications. But Minoru Kanisha went on and did something very big that you all must have seen before, I guess. Can anybody name it? So Minoru is the brain, the driver, and whatever you call it, behind KEGG. the Kyoto Encyclopedia for the pathways. So essentially that is the thing that contains all the pathway annotations. Minoru Kanegisha. And that is one of his early papers. Again, the claim is that amino acid composition correlates with sub-cellular localization. They show that it works. But do you actually believe the claim? And this is, well, is only relevant for the people who were not in the whiteboard lecture last week. So what I show here is a simple three-state system. And I can't remember the colors. Yes, blue is extracellular, green is nuclear, and red is cytoplasmic. So what you see is the clustering of those three sub-cellular localizations according to the two principal eigenvalues of the amino acid composition vectors. Okay, so again, I give it to you that this slide is not very good. But what you certainly, I believe, will trust when I tell you is that there is some separation here. Green, blue, red. Right? And you immediately understand that Kenta Nakai and Kanegisha could use that signal. Now, we discussed last week that most likely amino acid composition is not the irrelevant thing, but it should be the surface of the protein, right? Because only the surface sees differential. Now what you see here is co-residues. And now at least the blue and the red are completely overlapping. There's still some cluster in green here, but there's also green here, and there's also green here, and this is difficult to see. Let me inverse this. This is the surface residues. And you clearly now see that the clustering here is better than here. So that is completely the expectation. You see, in fact, that the surface is the thing that is the most important signal, and this signal exists because it comes from many residues on the surface, and they see the environment. Now, on a larger data set here, you see more clearly the clustering, and then you see some cases where, in fact, the blue here is right flat in the cytoplasmic, the extracellular protein is right in the cytoplasmic area. When we looked at the most extreme outliers, we found that they were glycosylated. So, you put a sugar on top of your molecule, and by putting a sugar onto something that is in the realm of not being charged, you add a charge. You change the surface composition. You change the surface feature. So you essentially have a very, very simple mechanism to make your protein appear or be happy in a different environment. Take a sugar off, take a sugar on, this thing lives in two different environments, happily, right? So it's right, looks like a cytoplasmic because in fact it has a sugar on it and it looks to the environment like that. Very convincing principle. And this, in fact, is the overall compositional difference on the surface between nuclear, cytoplasmic, and cellular. And you see there is a clear difference. And you could use that for some simple prediction algorithm, but then ideally you want to put that into machine learning. In order to do that, you have to predict solvent accessibility, and that can be done relatively easily, and we discussed it last semester by a simple neural network. We can use solvent accessibility to infer function, and has anybody of you heard of conserve? So conserve, essentially all it does, it comes from the group of Nirbental, as the last author here. Glaser is the first, but the idea really comes from Nirbental. Originally, all they did is they looked at the surface of a protein in a three-dimensional structure and mapped amino acid conservation upon it. So conserved is dark red, variable is dark blue, and you see the most conserved part here is in fact exactly the binding site. That makes sense. Conserve is simply the tool that allows you to visualize easily something that is extremely important. In fact, I believe it was such a breakthrough and continues to be such a helpful tool, simply because they were the first to mix two words. The people who looked at amino acid alignments or sequences, and the people who looked at structures, and immediately fits together. Previously, people did that on two different screens, but nothing beats the visual way this is represented. Consec is, so replacing surf by sec, you get something that works or gives you some preferences from sequence alone. So this only works on a structure, once you have the coordinates. Now back again to how we can do predictions of sub-cellular localization. We want to use this principle to have sub-cellular localization prediction, and that again was done by Rajesh Nair. And so when he read it, what I showed you before, surface, all amino acids, different secondary structure combined, he saw that there was some signal, but a fairly weak one. Are you all familiar enough with neural networks to understand how they work? What's the difference between an SVM and a neural network? So essentially, I'm taking those because those are the most extreme examples that simply take two vectors and project those vectors into a different dimensional space and separate points by introducing some hyperplanes in that higher dimensional space. That's why they are similar. Just what I said for the neural network, so you have an input, you have an output, and you essentially project two vectors upon each other. And what you can do is you can introduce a line if you had without a hidden layer. With a hidden layer, you can essentially allow yourself more lines. That's the way it does. The operation is very simple. You sum up the signals here, and you have a sigmoid function to decide where the output lies. And then you reduce this by going down the error function. There's a simple error function here. Going downhill, adjust the coordinates according to downhill. There's this phase of overtraining. This is not neural network specific. That's true everywhere. Anyway. And that, by the way, is just one example in which you can use a neural network to predict sub-cellular localization. So you have, for instance, one way of doing it, you have one that has only sequence input, only solvent exposed composition, and then sequence and secondary structure, and predict sub-cellular localization, nuclear or not nuclear. Each one of these integrate in a higher level. That again is pairwise nuclear, not nuclear. That's one way of doing it. And in contrast to that, the SVM does in fact pretty much do the same thing. That's the original problem that is converted into a transformed problem in a higher dimensional space, finds the line between. The difference now is that where the neural network optimizes the separation of red and green in this particular example, the SVM picks a few points that are closest to each other. So in this particular case, would ignore those green points here, would ignore those red points, would say these are closest to each other. In fact, because they are close to each other, this is what matters most for the distinction. These are trivial points here. The complication is here. So let's optimize the line so that it can exactly cut between the ones that are closest to each other, that are most difficult to classify. Okay? Now, this has two impacts in terms of for what type of problem you use neural networks as opposed to for what type of problem you use SVMs. The major difference between the two. The math underneath is very similar.
I think in the case of S4M, you have always a solution.
No, in none of the two cases actually. So let me just give you an example where you come in with an input feature, right? And if the input feature, if you put in the identical sequence, and say one case you try to train is nuclear, and one you try to train cytoplasmic, you already don't have a solution, neither for an SVM nor for a neural network. So simply put, if the problem cannot be separated, then none of these two devices will separate it. And that ultimately has to do only with the input space. So by the input space of the identical sequence, in this particular case, I constructed an example where most likely the experiment either made a mistake or, again, remember, in different types of cells we have different proteins may be expressed in different compartments because they are in different environments. or they have different binding partners, or so the moonlighting proteins are examples for that. But again, so this is the type of problem that the system cannot separate. No, but in terms of you, what type of problem for what type of problem the SVM is better, and for what type of problem the neural network is better? What type of data? Any idea? Well, you see, somehow this image here explains it both. Since you throw away so much data, implies that you can live with less data. So SVMs work with much fewer data than neural networks, because they zoom into the region that is most important to distinguish. That's the strength. Now the weakness is also clear, and essentially is what I told you already. So let's assume that one of these points were mislabeled. So if you have mistakes in your data, then this will be most sensitive to that, because it will try all the time to separate out points that are incorrectly labeled. While the neural network finds an average over everything, so mistakes in the data, the neural network tolerates. The SVM doesn't, because it zooms in exactly into that. There are some functions which allows the SVM to disregard some fraction of the data. So those are parameters you can play with. But nevertheless, at the end of the day, dirty data is a problem for an SVM. Little data is a problem for a neural network. Those are essentially the simplest way to define when to use one over when to use the other. So in environments where you have ample data, go as a neural network, you're safer. In an environment where you don't, you have typically to do SVM. Be very careful that you find ways to pre-filter the data. So again, what I told you implicitly in passing before was that we have binary ways of classifying this. So I start with my protein and I have one SVM or neural network that distinguishes nuclei from everything else. What else could I do? Well, let's get back to the first question. Or another question. Since I construct this in terms of pairs, so I build one model nuclei against the word cytoplasmic versus extracellular mitochondrial, that allows me to go back to the biology and say, in biology, I have particular sets of pathways. So the secretory pathway, the gated nucleus, and I know this is biology. I can take that view and put that into my system, right? The way to do that is simply by deciding the first decision that I make is secretory pathway or not. And then I go further on in detail. So in this sense, I sort of built my model here on the way I group these pairwise things onto the pathways I observe in the cell. I could do it other ways. What else could you do? Any other way in which you could introduce a hierarchy? So if you don't introduce a hierarchy, let me tell you that part of the issue. Now, say this one here predicts nuclear, and this one predicts cytoplasmic. What do you do then? You may be inclined to say, well, if nuclear has the higher probability of nuclear, then you pick that one. The problem with that answer is that the data here may be very different in terms of consistency from the data that you trained this one with, and that may be the only reason why you have more consistency. In fact, that happens to be the case. So you could do it, but you would make many mistakes in doing that. That's why you sort of cry for a hierarchy. Now, I showed you the biological hierarchy. Just looking at it, what else could you think about? I leave it to you to, in fact, you have these pairs a certain level, and I show you the solution that Rajesh did. What other solution could you do? Or what other criterion could you invent? You have a bunch of pairwise classifiers. In this particular case, at the end of the day, we distinguish 5 different states. And we have 4 different classifiers. How can you invent a hierarchy? No? Come on. It's four different ways of arranging things. You could just try them all out and see what is best. Right? Which is something that we didn't think about at the time. Because you know how it is. Days are short. And Rajesh just got everything done. And then we published it. Had worked at that point on this issue for many years already. We'd started with something very different. At the moment we found the solution here, 2005, he really wanted to defend. As in the US, he at that point was 6 years with me. Or 7 even. So he had done his PhD thesis for 7 years and he had enough. By birdie, right? So the funny thing is I later found out that somebody in fact used the same data and found the model that gave the highest accuracy. Okay? And that's the one you see. Total coincidence. So we only did what we believe made biological sense. And then he tried one other thing that he had thought about, which essentially he looked at the thing that gave the highest pairwise signal up here. And he found out that this one was better than his first way of doing it, so he was happy with that one. That was the only reason. But it turned out from an optimization of all possible paths, this in fact was the best for more states than the ones I show you. Now, the great aspect of this sort of taking it into a tree, into a hierarchy, is that for every decision here, you see, in fact, you can make this SVM specific to that particular problem. For the secretary pathway, we already know the internal composition is important. Signal P is important here. It's not going to be the same for the others. So I can have this model look at very different input features, right? And this is also, that implies that the subsequent levels here, they do not see all the data anymore. They here, for instance, see only intracellular proteins. The secretary stuff is out for training that device. So that is, and the other thing that is clear is the further you go down, the more similar the classes become, and the more difficult it will be to separate them. You see the prediction accuracy here on the highest level is in fact higher than on further down levels. And that sort of gives the thing as an overview. Now, the pro of this model is that you have better discrimination. You can do things that are specific to that particular path. The disadvantage is, once you incorrectly classified your protein into the secretary pathway, although it's not, you cannot recover from that. We have the same problem. You cannot say at the end of the day the alternative path would have given down at the leaf something higher, because you can no longer compare these things. And there's no way out of that problem, okay? And there's really no way to do this in a way of combining both worlds, or no, sorry, we haven't found any way to just get rid of that. So find a way out of mistakes. Maybe we could. Or use best of both worlds in some sense. Later the system got a little bit more detailed, and Tanya developed something that is much, much better than this, and she's going to talk about that on Thursday. Some other methods to predict sub-cellular localization. So publicly available methods are NNPSL, subloc, psort, signal P, we talked about already, and we will give you some more. Let's begin with psort. And that again, now here we have Minoru Kanesha, the man who brought us KEGG. Again, immensely visible, but the impact, I would argue, is way beyond what this one seems to be indicating. KEGG is just so immensely important. That's Kenta Nakai. Again, he did psort, but he also established first that amino acid composition. This paper I showed you before is relevant for sub-cellular localization prediction. Again, very, very successful scientist. In the beginning, the idea really was to have an expert system that begins on this assumption that amino acid composition differs, and pull in extra information. The way they pulled in extra information is that in some cases they had cleavage sites, they had motif information. And they simply, whenever they have that information, you use that. Then you have a transmembrane signal, then you have amino acid composition. You combine whatever information you have, and you put it onto something like a decision tree, if you want. Right? This is pre-machine learning, 91. That's the beginning of PSORT. Paul Horton joined the group and in fact brought it into a more generic machine learning kind of program that looks at that is a better computer science job to connect all the pieces. The PSORT2. A PSORT2 essentially uses whatever feature it can get there in terms of sequence similarity, in terms of everything else, to predict sub-cellular localization. The latest version of PSORT2 is called Wolf PSORT, and that is in fact the quote for Wolf PSORT. And this is essentially about important updates. And I argue most likely this is the most used method for predicting sub-cellular localization, other than signal peptide. Target P, we use target P, signal P are specific to particular tasks. PSORT, Wolf PSORT are the ones that are sort of generalists in some sense. Here's Sherlock. Sherlock, and that is from Kai, with Oliver Kohlbacher, uses in fact text mining plus prediction. So it combines in some sense what I showed you before, the keywords, but now keywords text mining on the level of abstracts, on the level of PubMed. So that's a different level. So what I described to you before, and you were not in the room, but the idea of a controlled vocabulary, so the way we compile these vectors, that was only possible because Swisspot is a controlled vocabulary. On free text, these vector entropy assignment of sub-cellular localization could not have been done. Because then the vector immediately would get much too long and you cannot compile the entire vector. Complicated issue. But so the way they do it essentially is they combine motif search, go phylogenetic information, epigenetic information, then various SVMs to predict sub-cellular localization, including the way of abstract level. Multilog also uses information about sequence motifs on a better level, and in fact sort of it blows up my alphabet of classes that I predict and blows up the complexity in the way this is combined. So we begin with this query sequence, and then we have different engines here that look at different themes. Here are the motif searches, NLS is just one of those, and all of that then is combined in a set of SVMs that essentially look at a hierarchical settings, just like before. So from the way this is constructed, this is sort of in some sense the most advanced way of doing it. And Multilog 2 does the same, but it integrates phylogeny, so it looks at essentially profiles as opposed to, and we have seen before, the profiles clearly help. Importantly, again, comes from the group of Oliver Kohlbacher in Tübingen. Cello. Now, the trick of Cello really lies in the way they code. In the way they code the interm, they partition their protein into K-mers, segments of K-mers. They look at, again, E-motif type of motifs, so generic motifs, and they look at the sliding window. And they combine all of that into a method that is called Cello. So now I talked about a very coarse-grained view. The coarse-grained view, there's a nucleus. Nuclei have a substructure, right? There's a nuclear pore, there's a nuclear matrix, there are subnuclear compartments. And Mikhail Bodin developed a method that uses, he's one of the authors of MIME, the motif finder that we talked about before, and is one of the foremost experts on using Bayesian networks. And so he used Bayesian networks. This is just one way of sorting into subcellular compartments. And you're going to get this talk not from me, but from Mikhail, I assume, who is going to join us in December. So I'm going to skip this part here. And he's going to also talk much more about Bayesian networks and how they can be used for other topics. So, look at protein interactions and sub-cellular localization in this tool here that unfortunately is outdated, but I just want to present the idea to you. PNet. The idea was, let's just look at a bunch of protein interactions that have an annotation from experiments. Let's separate them according to the prediction of sub-cellular localization and others, such as transmembrane helix or the gene ontology number. And one way of looking at it is to simply ask, when I have different sub-cellular localizations, what's the probability to observe that two proteins from these two different proteins, sub-cellular localizations, are interacting? Green means higher than expected. Red means less than expected. Significant by to standard errors, or whatever our criterion was at the time. And this work mostly done by Jana Fran. And immediately see that there is a pattern that evolves from this, that you can now use to label the interactions that you observe. You can do two things. You can put this label, like a criterion, and you can simply take the bunch of interactions that you have and put it onto the simple view of organelle, nucleus, mitochondria, cytoplasm, membrane. So essentially, you take this, what used to be a cloud of experimental annotations, and you simply pull it apart in Cytoscape. So you map it to different regions, right? Again, since we predict that the protein is in the cytoplasm, we don't know where it moves, but that's not important. It's just these are nuclear proteins, and you see that you immediately get a new dimension for the annotation of protein interactions. And you immediately see how you can get a visual advantage. So the ranking that I showed you before is an aspect, but the other aspect really is that you now have a new way of displaying it. Janai looked at one particular example of the protein called Q9P2H0. That was here. And usually when this was an example of the Alzheimer's disease pathway. And in this particular disease pathway, I can't remember what the number of interactions was that he had at the time, but say there were 100. And before you look at this one here, most of you will immediately see that this means that it's a protein that doesn't have a name. That means that it's a protein for which we don't know the function. So when you go through a list of hundreds of pairs, you will always start from things you know. And you will clearly ignore this one here. By just looking at the sub-cellular localization, you suddenly see that this here is the crucial connection through that ER. So suddenly something becomes much more important. Simply by mapping it out, by having a relatively high accuracy resolution of sub-cellular localization prediction. Originally, I motivated why we want to predict sub-cellular localization because I told you it has something to do with function. Here we see it has something. It immediately buys you a very cheap way of achieving something that otherwise you could not have achieved. You would have always ignored that. Now suddenly it's clear this is an important protein to look at. In fact, after that, people began working on it, and it most likely now has a different name. Just one example how to find a way to use your tool and integrate it into something else and make it become more useful. So the prediction system to predict sub-cellular localization is still the idea that we have here means that we could in fact also predict the post-translational modifications. So N-terminal signal peptide is one post-translational modification because, I'm not entirely sure who of the two asked that question, because in fact it is on the gene sequence, and it is in many cases mostly cleaved, so it's a post-translational modification and a very important one. And then there are proteasome cleavage site, proteolytic cleavage site, phosphorylation, lipid modification, and then O-glycosylation, and so forth and so on. So far, every one of these is typically defined by a machine learning device or by a local motif. So far, we do not have a machinery that in fact handles all post-translational modifications. And everybody who I know tried to do something like the generic system failed. So far, really, the individual best methods still take the day. Those are by far the most common post-translational modifications. There are many, many others, and they become more and more important as signals that regulate functional import, export, and important for the function in terms of the process of a protein. And many more prediction methods need to be developed. The problem is that the data is not easy to get by. This is just one. By now, I guess I should change that slide. I guess the best way to get at these is Google and Wikipedia at this point. I'm not sure whether that still is the best site. Exposé. So I rushed through 187 slides. I want you to remember every single letter and every single one of those, as you understand. That's not true. Tobias is not going to do the lecture on Thursday, as I said. Tanya is going to do that. And most likely, so she's going to give her development of an advanced method that we believe is by far the best that is available at the moment. And then I will talk next Tuesday about protein-protein interactions. And when I remember, so the first one here will be a whiteboard talk on Tuesday, not recorded. And the second one on next Thursday will just be the slides again. Thank you for your attention.